Today's video is on our skeletal muscle structure and physiology. Just FYI, I have a lot of notes written on the side over here. I'm not going to read them or talk about them necessarily, but everything that I say will be, spoken. And these notes are there for you to review and read when you're re-looking at these notes. So as review, because hopefully you remember this from human anatomy, we have our skeletal muscle, and surrounding our skeletal muscle, we have a piece of connective tissue called the epimysium. The epimysium covers the whole skeletal muscle, and if we take a cross-section of skeletal muscle, we have these bundles of fascicles. They are called fascicles. So we pull out one of these bundles. It's called a fascicle. And this fascicle is also covered with a connective tissue layer, which is called perimysium. Notice that in the fascicle, we have bundles here. We pull one of those out. This is one muscle fiber, which is one muscle cell. These are also covered with a connective tissue layer called endomysium. Endomysium, perimysium, and epimysium, the first one we spoke about, all extend beyond our skeletal muscle, and that's actually what makes up our tendon. Our tendon is our epimysium, perimysium, and endomysium. We can pull out one muscle fiber, as we can see here, and what we would see is that our muscle fiber is also made up of a bundle of fibers called myofibrils. And we are going to talk in more depth about these myofibrils. This is what muscle fiber looks like beneath the microscope. One really big characteristic that you can see is that it has these stripes called striations. It's not the only muscle type that has this though. But one thing that is special to skeletal muscle is that it is multinucleated. It is multinucleated. Notice that this is one muscle fiber. It's got a bunch of nuclei. And so our muscle fibers, our skeletal muscle fibers, are really big. They can be from our hip to our knee, right? They can extend the length of that. And so they are actually a collection of muscle stem cells that have fused together, and they are really specialized, which is why we'll see they have special names for their cell parts. Here is a closer look of a muscle fiber. You can see the myofibrils packed inside it. And then inside of that, we have myofilaments. So you can see these myofilaments that are part of the myofibrils. These are composed of thick and thin filaments. They're called thick and thin filaments because beneath the microscope, they look like thick lines and thin lines, and that's what gave rise to that name. If we can look closer and see here, you can see like a microscopic picture of it. You can literally see this thick and thin filaments, but we're going to look at this drawing just because it's easier. And what you can see is that we actually put names to the different bands that we can see. The purple bands here, the purple lines here are our thin filaments, which are made out of actin, troponin, and tropomyosin. And then the green ones here are our thick filaments. Notice that they're thicker, and they're made out of one protein called myosin. But when we look at them beneath the microscope, you can see these different banding, right? You can see like different banding patterns. Where they look really thick is called the A bands. And they look really thick because you can see both the thick and thin filaments beneath that area, like kind of right here. In the center of them, notice there's kind of a break-off point where there are no thin filaments. That's called the H zone, and you can kind of see that in that picture right there. So that's an area of only thick filaments. And then side to side, you have the I band, which is going to be an area of only thin filaments. And between them, there are these Z lines or Z discs. They're called that because they look like Z's. And they are where the actin attach. And so we define a sarcomere. It's written on the page actually. A sarcomere goes from Z line to Z line. And a sarcomere is actually what is going to shorten in length as we contract our muscles. So here's a closer look. We've got our A band and our H band in the middle, our I band where there's only thin filament, our Z disc in the middle, and then our Z disc to Z disc is what we call a sarcomere. Sarcomeres are going to be the contractile unit. It's actually going to be the thing that shortens in length. The thin filaments do not get shorter. The thick filaments do not get shorter, but they kind of slide across each other. And so we're going to talk about the sliding filament theory. Notice that the thick filament here, which is myosin, is attached to a filament called titin. Titin is a structural protein. It's attaching the Z disc and the myosin. It's attaching the myosin to the Z disc. It is elastic, which means it can recoil and go back to the original shape. So all these filaments that I've been talking about, actin, myosin, titin, they are all protein. They are all protein. This is why protein is so important to take in while you're trying to build your muscles. So in the sliding filament theory, our sarcomeres are going to shorten, but the thick band does not shorten, the thin band does not shorten. What actually happens is that the thick and thin filaments are going to slide over each other, which is why it's called the sliding filament theory. And notice that the I bands will get shorter. The I bands get shorter because what happens is the thin filament is going to overlap the thick filament. So the I bands get shorter and the H band gets shorter. The way that this happens is myosin, which is shown in green here, is really attracted to our actin, which is again shown in purple. Myosin really wants to bind with actin, and when they can, it's going to bind right away because it has a enzyme called ATPase. ATPase allows the myosin to take an ATP molecule and hydrolyze it and release a phosphate. And what that does is it kind of cocks it back and it's ready to go. It's cocked back, ready to go. As soon as there is a exposed binding site on actin, it's going to bind and cause a power stroke. This power stroke is actually what causes our muscle to shorten as it pulls the thin filament over the thick filament. Remember, they're going to overlap over each other. So let's walk through it from the top. At rest, notice that there is already a hydrolyzed ADP and phosphate here, so it is cocked back, ready to go. As soon as the binding site on actin is exposed, it is going to bind and it causes the power stroke right away. And it causes the thin filament to move over the thick filament, causing our sarcomeres to shorten. This is staying attached, by the way. This stays attached until there is a new ATP molecule to release it from contraction, which means that we need a new ATP molecule to relax our muscles. This is relevant. Maybe you've heard of rigor mortis. If you are aware of what rigor mortis is, It's something that happens about 4 to 6 hours after death, and it occurs until about 24 hours after death. But say if somebody were to die in a seated position and left there for 4 to 6 hours, their body would not be able to move. You would not be able to release the muscles from the way that they were contracted. So if somebody was sitting as they passed away, they would stay in that seated position until about 24 hours. That is because calcium, and we're going to talk about calcium in a little bit, calcium will be released, which is going to cause this action to happen. And so the cross bridge occurs, it's going to contract our muscle. And because this person's dead, they have no new ATP. They have no new ATP to release this contraction. And so their body stays contracted until there is tissue degradation after about 24 hours. And so rigor mortis is because of this mechanism here. Okay, how do we control this? Well, actin is really interested in binding with myosin, but there are some controlling proteins here. Tropomyosin, which is kind of like this rope structure, and then troponin. Tropomyosin and troponin are going to regulate, they're regulator proteins, they're going to regulate whether actin and myosin can bind. In relaxed muscle, tropomyosin is this rope. It is covering up the binding sites on actin so that myosin cannot bind to it. So this happens when calcium levels in the muscle fiber are low. When calcium levels are low, the muscle is at rest. Tropomyosin is covering up the myosin binding sites on actin. And then when calcium levels are high, calcium will bind to troponin, which are these regulatory proteins over here. Troponin controls where the tropomyosin rope goes. So troponin, when it binds to calcium, it moves the tropomyosin rope out of the way, and that exposes the myosin binding site, allowing for myosin to attach and for the cross bridge to occur and for contraction to occur. So contraction is controlled on the amount of calcium that is in the muscle fiber at the time. Calcium is controlled by a specialized structure in the muscle fiber called the sarcoplasmic reticulum. It is this network of green, it kind of looks like a gate along the length of the muscle fiber. This is a specialized version of the endoplasmic reticulum. It's called sarcoplasmic because sarco has to do with muscle, and it has a specialized function here. All it does here is store calcium. It is a calcium storage spot. And a lot of the calcium is in the terminal cistern, which is found on the end points right here. And notice that they're really close to these invaginations of the sarcolemma. The sarcolemma is the plasma membrane of our muscle fiber. Again, special names because they have a specialized function because muscle fibers are so different from our typical cell. These sarcolemma have these invaginations where they fold inward, and they are called T-tubules. T stands for transverse because they run on a transverse plane to the muscle fiber. So at any given time, the calcium is going to pump back into the sarcoplasmic reticulum. So when we want our muscles to contract, there's a lot of calcium in the space. It's going to bind to troponin and move that tropomyosin rope. When we are at relaxation, calcium levels are going to decrease because they're going to go back to the sarcoplasmic reticulum. Contraction occurs here because of activation from the nervous system. And so we have to look at the point at where the nervous system and the muscle meet, and that's called the neuromuscular junction. It is where we can see the axon terminal of one motor neuron and where it can innervate the muscle fiber. You can literally see this beneath a microscope. We're going to follow an action potential down the length of our motor neuron, and it's going to cause release of neurotransmitter from this terminal bouton. And we're talking about skeletal muscle here. And so the neurotransmitter it's going to use is acetylcholine. Acetylcholine will be received by the skeletal muscle fiber via nicotinic receptors, which means it'll open up sodium channels. And this is going to cause an action potential to occur along the length of our skeletal muscle fiber. And notice that there are voltage-gated channels here. And it's going to propagate down the length of the T-tubule. And remember that the T-tubule sits really closely to the terminal cistern of the sarcoplasmic reticulum. And when there is an action potential and a depolarization event, this is going to cause calcium to be released. Notice that calcium is released from the sarcoplasmic reticulum and then also into the sarcoplasm. This is called electromechanical release, and there's release on both sides. Calcium is going to bind to troponin, which will move that tropomyosin rope, allowing for actin to bind to myosin, allowing for that contraction to occur. So this is kind of the connecting link between excitation and contraction. Acetylcholine definitely starts the excitation in our skeletal muscle. Calcium is what begins the contraction. There was a slide earlier about Botox and what Botox does. Botox is going to inhibit the release of acetylcholine here. And so you cannot excite your skeletal muscle where you inject Botox. Botox at high levels can be life-threatening because you can imagine there are some life-threatening events if you were to inject so much Botox that it could travel in the body and it would impact, muscle, for example, your diaphragm, and you would not be able to breathe. So Botox is an inhibitory, it inhibits the release of acetylcholine, and that can be dangerous. So you definitely want to go to a reputable place if you're going to get Botox. For your muscles to relax, calcium is going to go back to the sarcoplasmic reticulum. It is put back into the sarcoplasmic reticulum by a calcium ATPase pump, and it's via active transport. So again, active ATP is needed for muscle relaxation as well as contraction. A motor unit is the motor neuron plus the muscle fibers that it innervates. And so notice in this image, this particular motor neuron might innervate 1, 2, 3 muscle fibers. So all of those would be, all those things put together would be a motor unit. When a motor neuron is activated, all the muscle fibers in its motor unit are going to contract. The larger the ratio of motor units, for example, if one motor neuron innervated 2,000 muscle fibers, that is going to be a lot less kind of like a dense or brunt force, you could say brunt force of control, not very fine control. But if you have motor units where one motor neuron is going to innervate maybe 100 muscle fibers, that would mean much finer motor control. And so there is a ratio here. The smaller the ratio, the more finely controlled.